is basically gone, and pouch 4 also is gone, but we're not really seeing any kind of duplication of any structures or any alteration or transformation or homeotic transformation at all in this particular mutant. So here's an example of a Hox gene that mutated. It did have an effect. It was essential for the embryo, but there was no homeotic transformation that occurred in this case. These are just some of the structures here in the area. Okay, so the next gene to look at was HOXA2, and that one is expressed a little bit more 3' on the chromosome. In the embryo, it's expressed also in the pharyngeal arches, but in a slightly more anterior region. And when you knock out HOXA2, you get a phenotype that affects pharyngeal arch number 2 and the pouches that are associated with that. So it is a little bit more anterior structures the more 3' you go in the genome, which again matches the expression pattern. But this also is not a homeotic transformation. So, and keep in mind that these are not the only Hox genes here, but we're just mutating these things one at a time. Okay, so each of the branchial arches is going to have some unique combination of Hox genes, and therefore you get the correct patterning of these structures. Okay, so what am I saying here? Okay, HOXA8. Okay, where was that guy? That was here. HOXA8 is quite a bit further down in the location on the genome. And now we're seeing a gene that happens to be expressed in the area where we see vertebrae in the thorax and trunk area. And so here's a little pattern of the vertebrae going from anterior to posterior for the mouse. So a certain number of vertebrae we typically find in the neck region. Then we get a certain characteristic number of thoracic vertebrae. Each one of the thoracic vertebrae has a pair of ribs associated with it. So that's sort of like the thoracic segment 2 in the fly always has a pair of wings. Well, these thoracic segments in the vertebrae, they always have a pair of ribs. Just the characteristic thing for thoracic vertebrae. Then we have some lumbar vertebrae. They're a slightly different shape. They're a little bit more thick, found more down in the trunk area. Sacral, this is the region of vertebrae where the pelvis is attached. And then caudal, a region that you would see vertebrae in the tail. So in the case of this HOXA8, both copies mutant now what you see is that this first lumbar vertebrae changed its identity and became a thoracic vertebrae. And part of the reason that we know that this is true, in addition to the shape and size of the vertebra itself, is that now this vertebrae started growing a rib on both sides, like all the thoracic vertebrae always do. They always have a rib. So this actually gives you a homeotic transformation because we've converted a lumbar vertebrae into a thoracic vertebrae and made an extra structure, which is the rib. Questions on that? Yeah.
So in the picture, you can tell that like each thoracic vertebrae sort of sprouts off a different pair of ribs, and so it almost looks like in this picture where the lumbar vertebrae is mutated that it's sort of following the same pattern as the rest of the thoracic vertebrae. Does that mean it was specifically mutated with that thoracic vertebrae that's just above it, or does that mean that there's something else that controls how long the ribs grow?
I see. How long the rib grows. I would say that each of these thoracic vertebrae has in life experienced a different combination of Hox genes.
Okay.
So the Hox code hypothesis is applying all the way along here. And the Hox codes is going to influence what the shape of the ribs are for say 2. So in this lumbar vertebrae, it got converted to look like the most posterior thoracic vertebrae. And so it did get a pair of ribs. And that rib might not be fully wild type sized, because it's not a totally clean situation here. All we've done is mutate one gene.
Okay.
But you can definitely tell it's a rib, and you can definitely qualify or score this as being homeotic because of that. Other thoughts or questions? Okay, so our answer was yes, you could get a homeotic transformation in vertebrae. And also, this just reemphasizes the theme that each one of these vertebrae have a different combination of Hox genes and that you can play with this code a little bit. So when this identity was changed, and you'll see this theme too, if you start looking at many of the Hox gene mutants, the new character that is taken on is usually a little bit more anterior in nature. So they tend to have this phenotype that reflects structures that are more close to the head, as opposed to going the other way of developing more posterior-like structures. And interestingly, that was kind of the same theme we saw for flies as well. It's a thoracic segment 3. When it changed, it took on a character of the more anterior-like segment, the thoracic segment 2, with the pair of wings. So that was an anterior horn transformation. And this partly has to do with just which combination of Hox genes are expressed where in the embryo. Okay, so we know we can get homeotic transformation. We know we have combinations of Hox genes acting together. And we know we have these paralogs due to the 4 sets of Hox genes in vertebrates. And so, coming back to this question of whether or not the paralogs share any function in and among their little group, the only way to really answer it would be to knock out all of the members of the paralogous group and see what happens. Because let's say they each have, 3 functions and one of those functions is in common. If you mutate them one at a time, there would always be 2 other genes around that are fully functional and could accomplish that common function. So you're not going to see a phenotype associated with that. You would only see a phenotype for a common function if you now obliterate anybody that can do that function. So we're talking here about making a triple, knocking out 6 genes, so 2 copies of 3 genes in the organism, and then studying what the phenotype might be. So this gene here is relatively far down the length of the chromosome, and it is involved in patterning structures on the trunk and tail. And so just conceptually, the way that you could create a mouse that's going to have this genotype, it'll be mutant for HOX10A, both copies, HOX10C, both copies, and HOX10D, both copies. This represents all of the paralogs. There is no HOX-B to HOX10D. Then you can look at what's the phenotype of this mouse. So what you do is you actually first have to go in and do targeted mutagenesis and make your mice that are mutant for each of these different genes. And then you breed them together until you can finally get your embryos that have all 6 copies of mutant genes for these 3, and then you examine what the phenotypes are. And I want to mention Mario Capecchi because he is Mr. Mouse Knockout. He has a huge group in University of Utah, and they've just been going through one at a time, knocking out all these genes, these Hox genes in mice, and studying the phenotypes, and then even get into these fancy experiments where you get triple mutants and double mutants and see what the phenotype is. So what is the phenotype? The phenotype has to do with ribs. So you get a homeotic transformation, and this time it's going to occur on a massive scale. So your wild-type skeleton looks like that top diagram there. So a certain discrete number of thoracic vertebrae, each with a pair of ribs. Then a certain discrete number of lumbar vertebrae. The sacral vertebrae, you can recognize them because they also have a little appendage associated with them, that sort of winged little phalangeal structure here, right in there. And then the caudal vertebrae, which have no appendages associated with them. And so when you knock out all 3 of these genes, now you're going to get ribs that go all the way down. So all of the lumbar vertebrae have ribs, and even the sacral vertebrae have ribs. And they're not, astoundingly perfect ribs, but they are definitely rib-like in character. And that would qualify as being a homeotic transformation to a thoracic-like situation all the way down the axes of the body. And so this is kind of a surprising result, because if you think about what is wrong in the mutant and you infer back to what that normal wild type function should be, so what is the function that we're missing here? The function that we're missing is basically rib repression. So each of these three genes has the wild type job of repressing ribs in the lumbar and sacral regions, because we don't want ribs to grow there. If you knock out all those genes, there's no longer a protein around who could accomplish that goal, and so ribs sprout out. And it's a little bit of kind of a, for humans, I think, a reverse psychology thinking, because we think that you should have to do something active to build a rib, not that you should basically have to be preventing ribs from growing. But you know, biology is like that. It does things its own way, and so it turns out that you do actually have to proactively repress rib growth in the embryo. Yeah.
So earlier you said that generally speaking, things act more anterior than they are mutated. And is it therefore true that things that are more posterior tend to be more repressed in nature?
Okay.
By repressing genes, and then that way if you're knocking them out, you're actually getting expression of the anterior?
Okay, so this is an interesting question. She's tying together the idea that we have individual mutants generally mutate to a more anterior character, and the idea that now some things are repressed, and do those two concepts correlate? And I think I would say the answer is no. So, you know, these would qualify as an anterior-like transformation, so that theme is upheld. And it just so happens that this particular Hox function has to do with repression of ribs. Other Hox gene functions might have a more proactive role, where they now build something unique for the first time. So I would not say it's a general theme to repress things the more posterior you go. Okay, so now if you had mutated only one of these three genes, then the other two would still be present and they could accomplish this function, so you're not going to see this phenotype. In fact, out of the 6 copies of 3 genes, if only one of them is remaining in the wild type situation, that protein will be enough to repress rib development. And so there's this huge amount of overlap or redundancy among these 3 genes for this particular gene function. And that is a theme that you can see for the paralogous genes, is genetic redundancy. So paralogous genes, they each have, you know, their own task list in life, but there are going to be some of those tasks that are in common among all the paralog members. And so the only way that you could see that phenotypically is then to knock out every single paralog that has that ability to do that job. Does that make sense? Okay. So this is the last slide, and it's comparing here a chick and a mouse in the anterior axes of these things. So in these 2 species, we have basically the same sorts of classes of vertebrae: cervical, thoracic, lumbar, sacral, and caudal. And we have different numbers of vertebrae in each of those classes, and this then reflects the appropriate morphology for the different species. So chicks, they have quite a bit longer necks, apparently, than mice do, if you start thinking about their skeletons and how that's laid out. Mice have a little, you know, longer chest cavity with more ribs than do chickens. And so then the colors are indicating different types of vertebrae, but then we see Hox genes here, 5, 6, 9, and 10, you know, getting larger in numbers to get more posterior. And the domains of expression for these Hox genes have been shifted from one species to the next. And if you think of this whole paradigm of Hox genes having jobs to do with specifying identity, and you change the width or the placement of this Hox combinations, you could then change the readout that you get for body structure down the line. And I think it's just really interesting to think about this from a comparative anatomy point of view, because it seems to make sense to explain why some of these structures have changed over evolutionary time and how the body plans, you know, if you look at the elements, they're there. Everybody has the cervical, thoracic, lumbar. It's just that we tweak this for different organisms. Questions or thoughts? Okay, we are done, and we'll see you tomorrow.
Yay.